Hi everyone, this is the second mini lecture in Chapter 13. We're going to be looking at the development of lymphocyte diversity, so the development of B cells and T cells. We're going to start with the B cell maturation, which occurs in certain bone marrow sites. Remember, B cell bone. These bone marrow sites harbor stromal cells in humans, and these stromal cells actually provide the chemical signals that will initiate the B cell development. Once these have developed, these naive lymphocytes, and they're referred to as naive because they have not yet been exposed to the unique antigen that they will recognize. These naive lymphocytes circulate in the blood, and they go to specific sites in lymph nodes, the spleen, and gall. When we're looking at T cells, remember T cells mature in the thymus gland, and the development of the receptors is directed by the thymus gland and its hormones. So there's a number of different T cell receptors that you will learn about in this chapter. There are the CD3 receptors, and these surround the T cell receptor and assist it in binding. There are also the CD4 receptors, and these are accessory receptor proteins, and they bind to that MHC2 molecule, which you should remember from my last lecture. And then we have CD8 receptors in the T cells that are found on cytotoxic T cells, which I'll talk a little bit more about in a later lecture. But the cytotoxic T cells will have these CD8 receptors that bind to MHC1 molecules. So these are different parts of the development of that T cell maturation. So if we look at the surface of a B cell and a T cell, you'll see a little bit of a difference here. We see that this is the B cell on the left, and it has that antigen binding site here. Now when we look at the T cell, we have an antigen binding site here, but you'll see it has these helper receptor cells near it, the CD4 and so on. So they're a little bit different looking. And when we talk about B and T cells, it's always good to know some of the differences between the two. So we already know the site of maturation. B cells mature in the bone marrow, T cells in the thymus. And then specific surface markers, things that are markers for these cells. You have immunoglobulin, which we'll talk about a little bit more when I go into B cells. And we have the T cell receptor, and there's several of those CD molecules that I just went over. And then how are they found in the blood, in circulation? There are low numbers of B cells. That's because they generally reside in the lymph organs. And there are high numbers of the T cells. The receptors for the antigens, there's the B cell receptor, obviously, and the T cell receptor. And then we talk about a couple of other things, the distribution in the lymphatic organs, where they're found in those lymphatic organs. And then with the antigen, do they require the presentation of MHC? And in this case, only the T cells do. And then once they're stimulated, what actually happens? So when a B cell is stimulated, we get plasma cells and memory cells. When T cells are activated or stimulated, we get several types of activated T cells and memory cells. We'll go into these as we go further into this chapter. So what is the general functions of each of these? The B cell's general function is to produce antibodies, which can inactivate, neutralize, and target these foreign antigens. The T cells, when they are activated, they help other immune cells. They can also suppress or kill abnormal cells. They can mediate hypersensitivity, and then they make cytokines, which also aid in that immune response. So how do we get this diversity of our lymphatic cells? How do we get so that those B and T cells can recognize all these unique antigens? And we're going to talk a little bit about that. So by the time these B and T cells reach these lymph tissues, the lymphoid tissues, each of these cells is equipped to respond to one unique antigen. And that diversity is generated by a rearrangement of the gene segment of the receptors, the receptor portion of the T and B cells. And every possible recombination occurs. So there is an enormous assortment of lymphocytes. As a matter of fact, it's estimated that each human can produce antibodies that have 10 trillion different specificities. So think about how big that is. We should be able to recognize any foreign agent that comes into our body just by the nature of this diversity. So when we talk about this, in the B cell receptor, that is actually the immunoglobulin molecule. That's the actual receptor for the antigen. And it's a large glycoprotein molecule, and it is secreted, when it's secreted, it is an antibody. And the immunoglobulin structure has two main areas. We have that antigen binding site. That's actually the part of the immunoglobulin that is going to bind to that antigen. And it's very variable, obviously, because it has to fit a wide range of antigens. And we call this the variable regions portion of that, which is areas of extreme versatility from one immunoglobulin structure to another. And this is a good view of what this looks like. We have a gene that encodes these variable regions, and different RNA transcripts will come from that same gene. And what we end up with is we have this very variable region that is the part that can bind to an antigen. And then we have what is called the constant region in these B cell immunoglobulin that does not have very much variability because it's not the portion looking to bind an antigen. So when we look at this, how do we select which ones we're going to keep or which ones we're going to make more copies of? So the mechanism of this is activated for both B or T cells by an incoming antigen. So because that lymphocyte specificity is pre-programmed, there is existing genetic makeup before an antigen has ever entered the tissue, right? We've already made this lymphocyte with this particular receptor before that antigens even come in. And each of these genetically distinct lymphocyte will express only that one specificity, so only bind with one type of antigen. And what ends up happening is if we then are exposed to a particular antigen and it gets bound by either a B or T cell, we are going to get clonal expansion of that, so rapid multiplication of that particular one when it's been activated by an antigen. So when might we not want to have extra of these T or B cells or have a particular one? One of the ways that we want to control these T or B cells is because there is such huge diversity, there's a good chance that we're going to produce some that will actually bind or have a receptor for self cells, cells that occur in our body. But we don't want to have an immune response to our self cells because that can cause potential damage. So what ends up happening is if we have a T or B cell that reacts to self, it will actually be removed through clonal deletion, and then we won't have that potential damage occurring. The basis of immune tolerance or tolerance to self is dependent on clonal deletion. And part of what we think with autoimmunity when we have an issue where our immune system is attacking ourselves is probably that there has been a loss of immune tolerance through the survival of some of these forbidden clones or clones that recognize self. So the last thing I want to put in this lecture is table 13.2, and this actually shows you a clear picture of what happens with that clonal selection and then the expansion of B and T cells. So you get this stem cell, we get this huge array of lymphocytes that have all different receptors for antigens, some that recognize self that we immediately eliminate. And so they're all just kind of waiting for an antigen to come in that they recognize. And when we're challenged with a particular antigen, it will bind with that receptor, and then we will have that proliferation of that particular lymphocyte that has a receptor for that sign of attack, that entry of the antigen. And we will get a large number of those in multiplication, and that is our immune response against a particular antigen. So we'll go a little bit more into the actual framework of B and T cells and also on antigen presenting in the next lectures.